Hi everyone, and welcome to our second video on Keep Unit 1 Biology, and here we will be discussing carbohydrates. So this will be a kind of long video, so without further ado, carbohydrates are essentially any molecule that contains the elements carbon, hydrogen, and oxygen, right? And the formula sort of goes like this. So you would have a certain amount of carbon, a certain amount of water molecules, so that the hydrogen and the oxygen kind of combine together to make a water molecule, and the Y means that you could have a certain amount multiplying by the water molecule, right? But that's not really important. That's just the definition of a carbohydrate, right? Now, carbohydrates could be of many different types. We have monosaccharides or monomers, we have disaccharides, and we could have combinations of monosaccharides called polysaccharides, right? So in short, two monomers make a disaccharide, right? Now, the most famous example of monosaccharides is glucose, and we'll be talking a lot about glucose and its uses here. A famous disaccharide is sucrose. There's also fructose and galactose as well. And the important polysaccharides we'll be talking about are starch, cellulose, and even glycogen, right? So let's dive into glucose, our monomer, right? So there are two major forms of glucose, right? Alpha and beta. And you could actually use this to combine to make different polymers, right? So you could have a certain amount of alpha mixed with a certain amount of beta, and then a certain amount of beta mixed with a certain amount of alpha, and so you go, right? Now, glucose has many uses. It has a large amount of hydrogen ions, right? So hydrogen ions are actually used by mitochondria in respiration to generate energy in the form of ATP. Glucose could also be readily dissolved in water, right? And due to the large amount of OH groups, now any molecule with OH groups can dissolve in water and participate in the famous hydrogen bonding, and we know we like hydrogen bonding, right? So this is the general structure of a glucose molecule. Now, it may seem complex at first, but as you practice to draw it, you will get it, right? And it's actually very important that you learn to draw it, right? So now a glucose molecule, it has the formula C6H12O6. So your drawing needs to include all of those, right? Now, it's very easy to start off with the carbons first. All right, so it's a hexagon shape, right? You could see that. So when you're drawing it, you could draw out your hexagon first, right? And starting over here, you will have your first C, C1, and then you go around the ring, C2, C3, C4, C5, and then C5 branches off into C6 over here, right? Now you have your C's. Now each C has an OH molecule and a hydrogen molecule, each of them, right? And they sort of alternate going like this, but that is not really important. It does not matter how we draw the rest of them. I will show you which one matters very soon. Right, so each of them has a OH and a H, and then finally, here has the H, but the C5 instead of the OH actually bonds to another carbon, which has H2OH. Right, that's just something you need to learn of. And finally, we have the last oxygen molecule over here. Right, now these numbers here are actually very important. The order that I put them, that is how the order is, and you will see why you need to count the carbon atoms very soon. So that is the general structure of a glucose molecule. Now remember, I told you that there are two forms, alpha and beta. How do they differ? Very simple. So this is an alpha glucose molecule, this is a beta glucose molecule. The only difference is the orientation of the hydrogen and the OH on the first carbon atom. Okay, so in an alpha glucose, the OH molecule is on the bottom, while in a beta glucose, the OH molecule is on top. That is the only difference between alpha and beta glucose. Right. So let's take a look at a disaccharide, for example, maltose. So maltose is actually composed of two units of alpha glucose, right? And they are bonded by a special bond called an alpha 1,4 glycosidic bond. Very important terminology there to learn, right? So again, the alpha bond is formed when the OH is below the carbon atom on the bottom, right? And if it was a beta glycosidic bond, then the OH would be above the carbon atom, as we just saw, right? So these here are two glucose molecules combining together to make the maltose, right? Now let's look at this over here. Now in this molecule, this is the first carbon atom, while in the molecule on the right, this will be the first carbon. So let's see which carbons are used here. So this will be C4. Now notice we have the first C and the fourth C here. And what is the name of the bond? A 1,4 glycosidic bond. So that's where you get the number from, which carbon atoms participate in the bond. Now you could see that in an original molecule, there would be an OH here and an OH here. But these are no longer there. The H's are gone, and an O is also gone, and then we only have one oxygen here. So therefore, if two H's are gone, that's H2, and one of the O is missing, we remember we have the other O shared between them, that means that H2O in total is gone from the disaccharide. So water is lost as a byproduct. So what can we name as another name for this reaction? A dehydration reaction. Right, we could also call this reaction a dehydration because you have lost water. If you don't have water in your body, you become dehydrated. Same like this this formula here. Okay. Sucrose is another molecule, and it's a disaccharide formed by an alpha glucose and a beta fructose. Now you don't need to learn how to draw the fructose, okay? I just want to show you again how the bonding works. So this time it's an alpha 1,2 glycosidic bond. All right. It's not very important to draw this. You don't need to draw sucrose. You don't need to draw fructose. It's just another example, right? Now sucrose has a good property. It's actually unreactive, right? And that's why sugars are transported in plants in the form of sucrose. So it doesn't get, it doesn't be dissolved and stuff on the way to where it needs to go, right? It doesn't make sense that you lose some of the things you want to transport because of some random thing that dissolved there, right? So sucrose is unreactive and it could be used in transport, right? Okay, so let's get to the polysaccharides now, which are very important. So the first one we want to learn about is starch. So starch is made up of alpha glucose units, right? So you see how important these alphas and betas are. Alpha glucose units. Now starch isn't found in plants, we know this, right? And the starch grains in the plant cell, right? Now very important, each grain of starch contains amylose and amylopectin, right? So we'll be learning about amylose and amylopectin separately, and we will see how they differ because of the little bonds between them, right? Now amylose is found at the core of the grain, right? So if you had a starch grain here, amylose will be found in the middle, and it's surrounded by the amylopectin, okay? So let's take a look at amylose. So amylose consists of repeating units of alpha glucose, right, and linked by the same 1,4 glycosidic bonds, right? It's alpha glucose, right? So it's alpha 1,4 glycosidic bonds, and there's about 200 to 15 glucose units in a one molecule of amylose, right? So you see how many can combine together to make a polysaccharide, right? So this is how it looks here if you were to draw it, right? So it's really just glucose connecting to glucose, hundreds of them in the same 1,4 glycosidic bond that we learned the first time, right? So again, it's a dehydration reaction. All of them lose water there, right? Right, so very important is that the molecule is a straight kind of molecule, meaning that it has no branches. It doesn't branch off like a tree. It's not technically straight, it's actually a helix, which means that if you look at it, if you zoom it out, it will kind of look like that, right? In little curls, okay? I will show you a picture of it very soon. Right, so now it has OH groups here, right? Look at them. All the OH groups are on one side of the molecule, right? Now what happens between OH groups, as I told you, hydrogen bonding, right? Very important, hydrogen bonding. So if this OH kind of attracting that one, and this one attracting this one, it's going to pull the molecule closer together on one side rather than the other side. So what that means is it's kind of going to curve. It's going to make the thing curve like that, and it's going to curve until it starts to go into a helix shape. Okay, so one side becomes shorter than the other, and that's what gives it its curve or helical shape. Right, now that is very important. Right, so the helix makes it very compact. Right, so because it's spiraling like that, it's not straight out and taking up plenty space. Okay, it's also insoluble in water, right? Because all the OH groups are being attracted to each other. Okay, so it could be good use for storage as well. All right, now let's take a look at amylopectin. Amylopectin is a little bit different. So this one has branches like the tree, right? So 1,4 glycosidic bonds, just like amylose, but at the branches there's alpha 1,6 glycosidic bonds, right? And that's only occur at branches, and I'll show you what I mean. This one actually consists of thousands or even hundreds of thousands of glucose units, right? Now at every 20 or so glucose units, a branch will occur due to a 1,6 glycosidic bond. Okay, so here in this diagram, in this blue box here, we simply have the regular alpha 1,4 glycosidic bonds between carbon 1 and carbon 4. And then at every 20 or so, we are going to have a branch here. So at carbon 6, remember the carbon 6 is the one that is out of the ring in glucose, right? So at carbon 6, you're going to have an alpha 1,6 glycosidic bond. Meaning, let me see if I could draw one here for you. So I'm just going to draw this over here, right? Very, very quickly. So C1, C2, 3, 4, 5. So the branched one will be carbon 6. And you have a H2OH here. And you're going to have a glycosidic bond, meaning you're just going to bind to oxygen. And of course, you're losing your water as usual. And it's an alpha 1,6 glycosidic bond. So this is the 6th. That's the 6th for the 1,6 bond there. And the 1 is going to come from the carbon 1 of a regular glucose. Right, so C2, C3 go down the road there, right? So this is a regular glucose here, and this is the carbon 6 of the other glucose there. So this is an alpha 1,6 glycosidic bond, okay? And that is when it will branch off and continue along its merry way. And then one of this will branch off, maybe one of this will branch off, and you could see where we're going with that, right? Right, so this is just a diagrammatic representation of how they look. You could see in amylose, the alpha helical shape there. And then in amylopectin, you have the branched shape because of the alpha 1,6 glycosidic bond. Okay? All right. Now let's take a quick look at glycogen. So glycogen is another polysaccharide, right? So in glycogen, it's also made up of alpha 1,4 and 1,6 glycosidic bonds. And we have glycogen granules in the animal cells, right? So they're very similar to starch, except it's smaller, right? And because it's smaller, a lot more could fit in a given space, right? So again, its structure is similar to amylopectin because it's alpha 1,4 and alpha 1,6 glycosidic bond, but in this case, they are just more and shorter branches, right? So it occurs more frequently, and the branches are shorter, right? But essentially, it's the exact same way to draw as amylopectin, right? So it's basically amylopectin. Right, very important. You will have a lab on testing for glycogen, glucose, monosaccharides, polysaccharides, etc. So glycogen is present if you have a color change to a red-violet color when iodine is added, right? And this will be explained to you in your lab when you do it, right? There's no need for me to dive into this and take up more of your time, right? Now here in cellulose, which is the last polysaccharide we'll be doing, this one is a little bit different. So cellulose is a structural polysaccharide, whereas glycogen and starch are storage polysaccharides, right? So it helps with the structure of a plant, right? It needs to be strong. So it has extended chains which form cellulose fibers, right? Which add strength and dexterity. So cellulose uses beta glucose units, whereas starch and glycogen used alpha glucose units. All right, and each successive unit of glucose is actually flipped or rotated 180 degrees. Let me show you how that looks in a diagram. So this is your first glucose here. All right, remember it's beta. In beta, the OH is supposed to be on top there. Now all of these glucose molecules are beta glucose, but you would see now, well, look, this is the carbon 6 thing, which is always on top, where there's alpha or beta, right? So you could tell that it's actually, this is a beta glucose that is flipped on its head, right? And then the one successive is flipped back, and then it's flipped again, and so you go, right? So it's actually rotated 180 degrees. And what this does is it puts OH groups on both sides of the chain, right? You can have OH groups on both sides. So in comparison to your amylose, which had OH groups on only one side and it started to form a helix, because you have OH groups on the both sides, they will attract each other and have strong bonds on both sides of the chain, forming a very strong fiber that has a high tensile strength, right? So many hydrogen bonds give it a high tensile strength. Okay, so how it gets to a cellulose fiber is that 60 of the regular chains, so this is a chain here, right? This drawing over here. So that's one chain. So 60 of these chains would are held together to form something called a microfibril, right? And many of these microfibrils then join together to form a cellulose macrofibril, right? Or this is where you get the fiber from, okay? That's just a fun fact for you, right? Now, macrofibrils, which we have now, they are actually made even stronger by being held together by something called pectins and hemicelluloses. Right, these actually act like a cement between the bricks. Right, if you think of the macrofibrils as bricks, the pectins and the hemicelluloses, they help keep the fiber strong. Okay. So that is how cellulose actually carries out its function as being a structural polysaccharide. Because of the OH groups on both sides, hydrogen bonding occurs on both sides. Okay, as the main point here. And finally, I have this table here that shows starch versus glycogen versus cellulose. It is basically a summary of what we just discussed, right? In our next class, in the next video, we'll discuss lipids and proteins and so on, right? So before I finish, let me just zoom out on each page so you could probably take a picture, write your notes, take a screenshot, whatever you want. Okay, and I will catch you guys in the next video. Okay, you can take screenshots there, you could pause the video, you can do as you like, right? Okay, bye everyone.